Good evening, everyone. I welcome you all to today's session on transforming life science graduates into clinical SaaS programmers as part of the BioIT and Biotech webinar series. Established in 2019, Nyberman Bioinformatics has earned global recognition as a leading bioinformatics service firm driven by a distinct vision. The primary goal at Nyberman is to establish a comprehensive network that unites academia, industries, students, and active researchers worldwide in the vast fields of bioinformatics, biology, and related subjects. To achieve this, Nyberman Bioinformatics Europe was launched in 2021, providing exceptional training and services to individuals across the globe. With a diverse array of expertise, our team covers a wide range of disciplines, including omics, structural bioinformatics, computational drug design, molecular modeling, health informatics, AI, ML, and so on. Throughout our journey, we have successfully organized numerous impactful workshops and skill development programs in various bioinformatics domains. Additionally, our extensive portfolio boasts over 300 webinars, each drawing an average of 500 plus participants from France and abroad. At Nyberman Bioinformatics, we are committed to fostering collaborations and offering a variety of opportunities such as project outsourcing, consultation services, freelancing, and customized training programs. With a steadfast dedication to advancing the field of bioinformatics, we aim to empower individuals and facilitate meaningful contributions to the scientific community. I would like to introduce you all to the BioIT and the Biotech webinar series. So this series is held twice a month, which introduces the latest trends, discoveries, and methodologies in the biotechnology community and vice versa. This platform is very welcome to all of y'all who can share your knowledge. In today's session, y'all are also going to get a participation certificate. And for this, y'all have to follow certain steps to get your certificate. So towards the beginning, I request you all that during the webinar, y'all can either send a direct message through Instagram, and for non-Instagram users, y'all can send your request through LinkedIn to Nyberman Bioinformatics for the certification feedback link. Now, this feedback link can be availed during the webinar anytime between 14 to 15 hours, that is in Paris, and 6:30 p.m. to 7:30 p.m. IST. Also, the response time for the Instagram message requests shall be taken till 17 hours in Paris, and that is till 9:30 p.m. today. And also, we can say that the feedback link shall be active from 7:30 to 9:30 p.m. in IST and 15 hours to 17 hours in Paris. So I request you all to kindly adhere to these guidelines so that you all can all receive your participation certificate. Now, allow me to introduce our esteemed speaker for today, Manini Srivastava, who is a remarkable individual with a passion for statistical analysis and bioinformatics. Currently serving as a statistical analyst intern at Eli Lilly and Company in Bengaluru, Manini brings a wealth of experience and expertise to the field. Her dedication and commitment to her work has been evident throughout her professional journey, including her previous role as an intern at Aranya Biosciences Private Limited in Chennai. Manini holds a Master of Science degree in Bioinformatics and Applied Biotechnology from IBAB, where she's consistently displayed academic excellence from August to June. With a solid foundation built during her BTech in biotechnology, Manini has honed her skills and developed a profound understanding of statistical analysis in the clinical domain. Her valuable insights and expertise makes her a distinguished speaker, and I'm delighted to welcome her to share her knowledge with us today. Manini, all over to you.
Hi, so good evening, everybody. As Maya have introduced me already, my name is Manini Srivastav, and I hope what I'm going to speak today will help you all in deciding your career journey. Now, may I please know how many participants in this webinar are actually from biotech or pure life sciences, or is there anybody from pharmacy as well? It's all right if you can just raise a hand. There must be some icon here. So the people who are raising a hand are from biotech plus life sciences plus pharmacy. All right. So I'll start. So as for my introduction, which Maya has already provided in brief, I've completed my graduation from SRM, Chennai, in BTech in biotechnology. So I had completed my graduation in 2019. Uh, and during my graduation, I did a major project, which is generally a part of course curriculum. So that project I did was at a government council, and that project involved me to explore where I had to explore certain bioinformatics aspects and certain biostats aspects also. So with that project, with that flow, I started realizing that I'm actually interested in this field and I wanted to do more. So basically, I wanted some combination of biotech, bioinformatics, and stats. And after that major project, I went for an internship like NIT Bhopal. It was a 1 year of internship again, where I had worked with a professor in purely in bioinformatics. So he was not at all related with biotech. He was just related with bioinformatics and stats. With that internship, I have actually gained much knowledge, which I could again apply or think of that what I wanted to do. So basically, that project and that internship helped me a lot in diving into my interests and identifying what I wanted to do further. So I was applying for various roles again. So I was applying for masters as well, and in that journey, I had cleared a DBT BITP test and interview. It's like funded by DBT. They basically give try to give you an experience with certain companies. They have a tie up with. So I was again placed in Chennai. It was a startup company. So in that startup company was what I learned was very different. My role was to test their chemicals or cleaners that they were making. So it was a CRO. They would get services, and the team had to build their clinicals. So my role in a team was where we had to develop a disinfectant which was bioabsorbable. So that entire process was very different to me. It was a new learning, but unfortunately it had to stop because this was during pandemic. So I started in Jan 2020, and by March 2020, my again apprenticeship stopped because thanks to COVID-19 pandemic. And then I was again to back square one. But all of these journeys in some way helped me in realizing my interests. So again, I came across IBAB and its curriculum. They have a really good curriculum which amalgamates bioinformatics, stats, and biotechnology very well. So I did my master's from IBAB, and from there I got a placement, and now I'm a statistical analyst. So I would like to go into what exactly clinical research is. To understand stat analyst as a career, you need to exactly understand what exactly is clinical research or clinical trials per se. So clinical research is basically a study where a compound is identified for a particular disease, and you want to know if that compound will be effective and safe. If it's a novel compound, then obviously these tests are important. But if you're competing against some compound which is existing in the market, you need to compare also. So there are comparator studies as well. So I'll just dive into the basics first. So as you can see in the slide, there's a picture here. This is a preclinical phase. Preclinical phase as in where a lab or a team has been researching for some disease or for some compound, basically on cell lines. And from cell lines, they move to animals. I hope everybody understands cell lines because everybody is from biotech or life sciences background. And from there, they identify, okay, my compound is now working well in cell lines in animals. So maybe I can file an application to test its effect on humans. So this is So preclinical is all about the tests on cell lines and animals. For the company or for the sponsor to start phase 1 and phase 3 trials, here at this stage, an application has to be filed where you have to collect all the preclinical data, submit it to the regulatory authorities. And you have to prove that your compound is safe for use in humans as of now. So basically, we come into phase 1 trial if your application is accepted by the regulatory authorities. In phase 1 trial, the objective is completely to test whether the compound is safe in humans or not. So that's why you can see it's written here 20 to 80 participants, which means there's a small size of patients that are involved in these trials. So the sponsor basically tests different amount of dosage of the drug, and they check how much amount is toxic to a cohort. So it all starts with a cohort. Cohort is like a small number of people. So after the dosage has been decided, okay, this is my maximum dosage or maximum toxic dosage, they do not move further or a higher dosage. They generally come down. So if there are multiple dosage that have been given to a cohort, suppose they start with 1 mg, 2 mg, 3 mg, 4 mg, or 8 mg, and they find out that 8 mg is the maximum tolerated dose, or this is the dose where a toxicity has been seen. So they actually come down to 4 mg. 4 mg is like the safest dose that can be given to the participants. Also, to test the safety analysis or dosage in phase 1, only healthy volunteers are involved. Healthy volunteers which do not have a underlying clinical condition or any kind of medical condition. So this uh, dosage testing is also known as safety analysis, because I'm checking if the drug is safe or not. Now, in phase 1, if this has been done, you have analyzed, okay, my drug is working well now, it's safe for use in humans. We move on to phase 2. In phase 2, we check the efficacy of the drug. Efficacy means how much is it's effective to a particular cohort. And that's where you increase the number of patients. So here in phase 2, you can see in the picture, it's written 100 to 300 participants. Basically, the cohort size has increased. And from phase 2, they actually start enrolling some patients as well. Since the dosage has been decided, they just want to see if at this dosage, any patient is facing any adverse effect, adverse reaction or not. And how much is the drug efficient? So at phase 2, this is also known as efficacy analysis, basically. Now, after safety analysis and efficacy analysis has been done, this is all done by clinicians which are involved in clinical trials, investigators. Investigators are basically doctors which are there in the clinical trials. Now, after phase 1 and phase 2 have been completed, the trial moves on to phase 3. In phase 3 is also a longer duration with more number of participants, because in phase 3, you want to see a longer effect of the drug. Since safety and efficacy have been analyzed, you want to see if on a larger cohort and for a larger number of time, are there any more adverse reactions or adverse risks to patients? So here the sample size or the patient number are also increased. Here you can see 1,000 to 3,000 patients. So in this longer time, if there are adverse reactions, all these data will be collected in phase 3. Now, after phase 3, you can see in the picture, here it's written drug submitted for FDA approval. FDA approval, I'll come to that on the next slide, but I'll just tell you here that after phase 3, all the data that you have obtained in phase 1, phase 2, phase 3 is put together or integrated together and is analyzed and then submitted to FDA, which is a regulatory authority. So after this, FDA your scrutinizes the data, everything that you have done in the trial, to confirm that what you're claiming is actually there. So when a drug is approved by FDA after phase 3, it releases into the market. So after the drug has been released into the market, so after a drug has been released, the patients can take the drug. So the doctor suppose I'm a patient and I go to the doctor with some condition, and there has been a drug in market which is just approved, and the doctor prescribes the drug to me. So in this way, there is a larger number of cohort that is involved again, which is taking the drug. So this is like a post-marketing analysis in phase 4, where the drug people are taking number of drug, and you see if somebody is facing again any adverse risks or reactions or not. So this is how like phase 4 works. Also in phase 4, the sponsor is not like sponsor is a company which is funding or is doing all these trials, is not directly related to the data analysis. It's like the doctor who is giving back the review. So if I face some adverse reaction in phase 4, I report to the doctor, and the doctor tells this back to the company. Also, phase 4 goes for a very long time. It's like a post survey or after the drug has been shared to the market. I hope I was clear enough. I hope everybody understood what I just spoke. If everybody is not clear, please, you can ask me again. Okay. Now coming to the organizations that are involved in clinical research. So US FDA is one of the major organizations that's there, because every approval, be it a drug, be it a device, be it an injection or anything, has to go through FDA approval for use in patients worldwide, for marketing or like for releasing it into the market. Everything has to go through FDA approval. So whenever the trial starts or trial begins, there's a protocol or study design. Protocol is where the trial is like they will write a document where they see how a trial has to go through. What is the protocol? What's the method? Study design is basically how are they going to randomize patients. I'm sorry, I'll explain randomize. Randomize is where you have a number of patients, and then you decide which patient is going to get the actual treatment and which patient will going to get the placebo. All right. So, I'm sorry I didn't explain placebo. So placebo is like is to check if you're actually sorry. So placebo is like if you it's like a comparative analysis. In clinical trials, there's so some patients might get the treatment and some patient will get the placebo. Placebo is just to say just to check if the drug is actually working or it's the patient's brain is that is actually working. So before the trial has to start, the protocol is designed. The study design is there to check how many patients will get what and how many patients will get what. And then after the trial has been completed, a data collection happens. So C dash is again an organization which tells you of a standard way to collect data. And then there is something known as C disk. So remember FDA and CDISC. FDA is a regulatory authority which approves the drug, and CDISC is like a data standard which tells you how you have to send your data to a regulatory authority. Because there are many companies and many companies do their own trials. If there's no standard format or standard way, companies will send the data to FDA in their own understanding or own format. So it gets a little difficult for reviewers to scrutinize your data. That's why there are set standards. And the standards that are set is by CDISC. So remember CDISC, then there is C dash. C dash is where it tells you of the standard way to collect data from patients. Like during the trial, what patient faced what patient or which patient is getting what treatment. After getting this treatment, what happened to the patient? This is like data collection. And then after your data has been collected, you have to standardize it or you have to create it in a data set form or a table forms for the analysis. So for standardizing that data, there is something known as SDTM. SDTM is like standard data tabulation model, which tells you how you have to separate your data sets and how you have to build or how you have to tabulate it. And then after the data has been tabulated properly, it's in the set standards, it goes for analysis. Again, for analysis, there are set standards, which follows ADAM model. ADAM is like analysis data model. So these are various models which tells you of how you have to collect the data, how you have to put it together in a standard way, and how you have to analyze it. After everything has been done, it goes to the regulatory authority for the review. Now, yes, now, so we have collected the data, and now we have to put all the data together and analyze it. So for this data integration and data analysis, mostly companies or industries are using SAS. This is where SAS comes in picture. So I'll tell you why SAS. Oh, oh, I'm sorry, I forgot this slide. So FDA is a drug regulatory authority in US, but similarly, there are other drug regulatory authorities in other countries as well. So for India, it's CDSCO. So for everything to be marketed or be in India, it has to go through CDSCO as well. Similarly for Japan and Europe. Yeah, so I was on SAS, right? So why SAS? Okay. So SAS, because first of all, it's required by FDA. It's like a regulatory compliance. And then SAS provides a friendly user interface in which a person who doesn't have a prior programming experience, they can learn easily. They can identify where to go, what to do. Plus, it gives you easy statistical analysis, like calculating mean, median is comparatively easy in SAS. And putting data together and integrating it in SAS is also comparatively easy as in other languages. Now, there is one important point why SAS is important because it protects your sensitive clinical information. Now, why clinical information is sensitive is because suppose I'm a patient and I have enrolled for a trial. So my information is sensitive for the sponsor because it cannot leak. If it goes to it leaks or some other competitors see it, they might use the information wrongly to manipulate it. So my information as a patient is highly sensitive. It should not be changed ever, like ever, because it really affects the drug which is in study. So there are languages in which you can actually manipulate the data if somebody has. Yeah, so SAS doesn't allow a particular data or a data point to be manipulated. Suppose I have a I'm giving you a very simple example, my age. If my age is there in my database, it cannot be manipulated by anybody. This is how SAS protects it. It does not allow you to manipulate values. This is why SAS is being used widely in industries. Now coming to the next slide. What is SAS? Right. So I've embarked upon why SAS and now what is SAS? So SAS is basically a statistical software tool which was developed in 1966 in North Carolina State University by James Goodnight and Anthony Barr. So the initial version of SAS that they created was for basically data management and analytics. They had very small steps where they could calculate mean or median or merge something. But later on, SAS was developed and multiple analytics or analysis functions or tools had been included in SAS. And later it became SAS Institute. They removed the project from North Carolina State University and created something known as SAS Institute. You can, after the webinar, you can Google what is SAS Institute again. Now, what is SAS made of? Like SAS has something known as data steps, something known as procedure steps, and it has built-in functions. So data steps is where you create your data set. For example, in this picture, you can see there's something known as data example. So your data example will be your output data, which you are intending to create. And set example will be your input data, which you're using to create your output data. So this is something how data steps work in SAS. And then here you can see proc print. It's a very simple procedure step where it will print the data that has been output here. And here you can again see I've put a picture of the data step. first you create the data step, you build your data, the data has been so you collect the data. From that collected data, you create something known as standardized data. This is where the data is being created. And once the data is standardized, you analyze it through various proc steps. So this is your analysis, and this is the final output. Okay. I hope I'm clear until now. If still anybody has some doubt or some lack in understanding, please ask me. So I'm coming to how to learn SAS. How can you learn SAS? So for learning anything, like not just SAS, for anything, you need to understand its basics. You need to understand basics, understand how it works, and then practice is a major key. So in case of SAS, there is something known as PDV level. I hope I'm not diving too much into the technicalities, but I just wanted to introduce how you can do it. So PDV is like known as programming data vector. So in the previous example, I had showed you there is some input data which is used to create an output data. So consider that input data as this input raw data, which goes into the input buffer. There is something known as input buffer in PDV. So whatever is there in my input data, when I'm running that patch of code, it will go into the input buffer. And from input buffer, it will go to its memory level or in PDV. And then from PDV, it outputs. So this is like a backend of SAS. What I've explained here is an overview. There is much detail to it. So first, so if you start learning SAS, understand or start from the PDV level. And then you understand what's basic SAS. Basic SAS, I mean, where data steps, characters, variables, some functions, or size of their variables, how you can manipulate small things. For example, converting rows to columns or vice versa. This is known as basic SAS. And yes, obviously your proc functions. Proc functions are like the procedure steps. Procedure steps might include calculating mean or frequency or create a whole summary statistics. And after you've understood your basic level, your PDV level, basic SAS, and then you come into something known as macros. Macros are something in SAS which helps you to automate a process. For example, I have to repeatedly calculate a mean of a particular data. So instead of writing different proc mean statements or different data steps, I can use a macro to which will automatically create a means of a particular data. So it's like you're automating a process instead of doing it multiple times. Here I have listed some useful resources which you can refer. First is SAS documentation. If somebody really wants to learn SAS, I would advise them to actually go through and read SAS documentation because it's really clear and it's useful. And Lex Jansen, 9 to 5 SAS Listen Data are some websites where they have papers on every single step for SAS. These are some resources that you can actually use or refer while you're learning SAS. Then career prospects in clinical research, which was also a question from various participants in the template that I had seen. So the career prospects that I currently know of are statistical analyst, medical writer, and regulatory affairs. Statistical analyst, where you get the clinical data, put it into a standard format, analyze it, basically make the data ready for it to be scrutinized by regulatory authorities. Regulatory affairs is a job role where a team of regulatory authorities check, like in a company, they have to see if what you're working with is compliant with the standards that is there set by the regulatory authorities. Regulatory authorities team is responsible for doing everything that is set by the regulatory authorities. Or suppose FDA is there. So what rules FDA has put forward, they are responsible for be for working in compliance with those rules. And medical writer is a job role where you write protocol or study designs or decide study designs, which can go forward in clinical trials. And yeah, so we come to Q&A session now. If anybody has a question, they can ask, please. Or
Yeah, Manvi, I'll take that up. Thank you so much for that very informative session. And I'm pretty sure all our participants have definitely gained some really valuable insights that is gonna help them in their journey ahead. So before we proceed to the Q&A session, I would request the Naiberman team to kindly paste the attendance code in the chat. So everybody, please note that to get your certificate, you all have to send this attendance code to the Neighborman team, okay, via whatever platform that you all shall be communicating with them. And this code is available for both the Zoom as well as the YouTube live stream participants. So do check the chat, guys, and do take the attendance code. And once you all send this attendance code, then only the feedback link shall be shared with you guys. Okay, so this is to ensure that all of y'all do get the certificate. Okay, so kindly do check the chat screen, guys, and do copy paste the attendance code and keep. So, Manini, we have certain pre-selected questions. So first, I'll be going through them. And later on, we'll also be attending to certain live questions that we'll be taking. And towards the end, all the participants shall be free to sort of unmute yourself and speak to to Manini directly. So the very first question that we got is, could you share some practical advice for someone who's just starting in this field?
okay. So my practical advice for someone I'm considering in this field by the participant meant biotech or life sciences or something known as clinical research. Whoever has asked this question, can you please clarify what do you mean by this field? Okay, so I'll just answer on my assumptions. If you really have to uh, go to a clinical related field, I would like you to first analyze where your interest lies. If you're really interested, please go ahead, work on it. If I really want you all to understand that wherever you want to go, you need to see if you're actually interested in doing it. And then, based on your interest, understand what skills you require and work on those skills. And then see if you're working on those skills, if there is an opportunity, just grab the opportunity. For example, for having being a clinical analyst or a statistical analyst, SAS is an important skill in the industry. So I would suggest for somebody who wants to go towards this field, you learn SAS or work on it properly. Thank you.
Okay, yeah. The next question is on certain guidance on how to learn SAS programming, and also if there are any exams for the same.
Yeah, so this question I did cover in my presentation. I'll answer again. The guidance for learning SAS is you have to understand the basics properly. You can refer resources, and soon we'll also be launching a series, so you can attend that. And there are SAS certification courses. You can go check on the SAS Institute website, but they are a bit costly. So you can decide if you want to take them or not.
Okay. The third question that we have is that clinical SAS is having a boom these days, but why is it that freshers are not getting enough opportunities? So, what is your opinion on this?
Yeah, so my opinion on this is because there's a lack in the curriculum that is set by the colleges and between the industries. It's not like students are not capable of it, or if the industry is having a boom, then why are there no jobs? There are jobs. But at this stage, you need to find out at which college or which institute will you get that exposure to explore these kind of areas or these kind of fields. Like in India, I've basically seen there's a huge gap between the college curriculum syllabus and the industry requirements. So there's no question in the capabilities of the candidates. It's just the gap, which is there in my opinion.
Right, right. And the last pre-selected question was, what is something nobody told you about this job role or career, but should have told you prior?
So there's one thing that I was slight aware of, but didn't know when I started is like the huge responsibility that comes over you and no scope of mistakes. Because if I do some mistakes in my job, there might be repercussions which will not be good. So it'll be a huge loss for the company as well, for the patients as well. So it's something that comes over you as a responsibility. Everything else is okay, but it's something that comes is the responsibility.
Okay, Manini. Now we have some live questions as well on the Zoom chat. So one of the question is, can you give any example of biotechnology wherein it has involved SAS programming? So do you have any particular example?
Example of biotechnology where it involves SAS programming. Hmm.
Yes.
So I haven't come across where people use SAS in biotech, but yeah, you can obviously use SAS for statistical analysis. If you have some projects in your institute or in your college where it requires for you to do some analysis, sure, you can use SAS. But I've seen majorly people use R because it's most available. But you can also use SAS. I haven't come across anywhere, especially during my masters, where people were using SAS. Majorly people used R. So, but you can give it a try. There's something known as SAS on demand. It's a like a free SAS interface in which you can work on, like it's for academics only. I hope
Okay.
I answered the question, whoever asked. I hope I was clear.
Yes.
Yes, ma'am.
Thank you.
Participants, you all can also unmute yourself and ask questions directly to the speaker. And I shall also attend certain questions. Yes, so I guess Kashi has raised his hand. Yes, go ahead, Kashi.
Thanks, ma'am. Hi, ma'am. Thanks for the webinar. Ma'am, I just need to know this. Could you please explain what exactly the C disk, C dash, SDTM, and ADAM, in line with SAS, please?
Okay, so CDISC, CDAsh, SDTM, ADAM are not related to SAS. I'll repeat. CDISC is an organization which has set standards for data collection, data tabulation, and analysis. Am I clear till this point to you? Am I clear? I'll
Yes,
explain
ma'am.
again.
Yes, ma'am.
Okay. So CDISC is an organization, first of all. Under CDISC, there comes C dash. C dash is where it's like a guide which tells you of how you have to collect the data. And again, if you go to SGTM also is also a guide. It tells you how you have to put your data in a standard manner. And Adam is like it tells you how you have to analyze the data. So these are all guides. And whatever is written in these guides are done using SAS. Like in SGTM, something is written that you have to tabulate your data in this manner. So that particular thing which is written in the guide, I'll be doing using SAS. This is where SAS comes in picture. Am I clear?
Yes, ma'am.
Now, do you have follow-up questions?
No. So basically, the series or series is just an standard set of rules laid down. So by using that via SAS, we will do the tabulation or figures whatsoever maybe use it for the data analysis.
Right. CDISC is an
Okay.
organization, right?
Okay. Thank you, ma'am. Got it. Got it.
Okay, Mani. So the next, I think one pertinent question is by Tamogna. So what is the proper roadmap to learn SAS?
Proper roadmap to learn SAS. Okay, so I'll start again. See, anything that you have to learn, you have to understand the basics properly, like get into the basics. In case of SAS, it's PDV, like I explained, it's a programming data vector. So this programming data vector is how SAS works in the hindsight. Suppose I'm writing a statement in SAS, for example, A equal to B. So how this A equal to B is behind, or how it's printing A ka value if you have given something. So that you have to understand properly because it's an important concept in SAS, something known as PDV. programming data vector. After you understand PDV, you have to go through basic SAS. Basic SAS is as in how you write data steps. And in those data steps, how you assign variables and their particular values. Data steps, and there are procedure functions. Suppose you have a data, how can you analyze the data using some procedure functions? And yes, SAS has a lot of built-in functions, which makes it really easy. Suppose I have to find something. Suppose there's a variable name and I have to find something in that variable. There are a lot of functions which you can use in SAS. So understanding its basic PDV, basic SAS, which includes data step, proc steps, functions, and then comes advanced SAS, which is macros. So these three things, if you practice or you learn, you can have a good handle on SAS. And please do refer SAS documentation. It's very helpful.
The next question is by Shrutika. What are the skill sets that are required to go into clinical research?
As of now, I would say SAS is required to get into clinical research, but there are some companies which are transferring SAS to R. So you can work on R as well. Like it's still in the process. The difference between SAS and R is majorly lies in its functions. SAS has a lot of built-in functions. R does not have a lot of built-in functions. But companies are shifting towards R because the SAS license is a bit expensive. So you can work towards SAS and R as well.
Right. So I think there's one more question, and I think I'll take this as a second last. So the last question is that by Dr. Roman, if you could mention how much sample size will be needed according to the disease prevalence to conduct the first clinical trial using SAS. So basically the sample size, how will you calculate it using SAS?
Sample size. Okay, so first of all, sample size is determined when the study has been designed. So whenever a sponsor or a company is designing a study, they have certain objectives. According to those objectives, the sample size is determined. So it actually depends study to study and objective to objectives. General size for phase 1 is like a very small number of participants, maybe 80 to 100. And also the sample size, the sample tests, statistical tests are all decided by the statistician, which is involved in the study. So this generally depends study to study, but if you want to give you a number, I would say 80 to 100.
Okay. And probably the last question I shall ask, Manini. So, if you can, educate the participants regarding if whenever a life science graduate has to transition to the clinical research domain, what sort of companies can they apply to apart from Eli Lilly now that they're aware of it? And also, if you could, tell us a bit more about do we require any certain, basic experience or do we require any certificates so as to apply? So, what is your view on this?
Okay, so companies that you can apply are IQVIA. I've seen people working in IQVIA for the same posts. IQVIA is there, Novartis is there, Novo Nordisk is there, then GSK. GSK, yeah, which I have seen people working for the similar post are GSK, Novartis, Novo Nordisk, and what did I? Yeah, IQVIA, I said. Yeah, and if you have to have a prior experience, I don't think you need a prior experience being a fresher because if you go to a company, they might expect you to have a knowledge of little bit of SAS, but I haven't seen for at least entry level jobs you need an experience because when you join a company or at entry level for freshers, I'm saying this for freshers, when you join a company, they will at least train you for a week or a month or so. So they will train you in SAS and then they might tell you the certain standards that I just told you. So as per my experience, for entry-level jobs, I have not seen that you require a particular SAS certification or some kind of prior experience.
Okay, Manini. Thank you. Thank you so much for your wonderful insights once again. And it was really a very informative session.
Thank
And
you.
I think it covered all the basic aspects, for especially for candidates who are in the life science domain and then they want to transition into the clinical research domain. Right. So thank you, Manini, for your time and also for all the participants. And before I end this session, I would also like to take this opportunity to announce that there will be a short course on SAS programming that will be coming up, and most probably it will be hosted in the months of January or Feb. So I request you all to stay updated regarding this. Do check out the Nyberman website or the LinkedIn platform or any other social media platform for further updates. And thank you, everybody.